Hello and welcome to my channel, Sam Biology Class. Friends, today we'll discuss about the biofertilizer Azolla and Anabaena. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Azolla is an aquatic form that floats on the surface of freshwater pond, river, lakes, streams and flooded fields. It is found in tropical and subtropical regions of the world. The plant body is typically 1 and 2.5 cm in diameter. However, it can reach the maximum size of 15 cm or more. Azolla is related to other aquatic taxa such as Salvinia and Marsillia. The sporophyte of Azolla is highly reduced. It is differentiated into root, stem and leaves. The stem is often called as rhizome, and the roots which arise from the rhizome remain suspended in water. The upper surface of the stem is covered with small scale leaves that are compactly arranged. The leaves are bilobed. The dorsal lobe is aerial and green. On the other hand, the ventral lobe is colorless and remains completely submerged in water. The dorsal lobe contains specialized mucilous field leaf cavities which contains the nitrogen-fixing bacteria Anabaena azollae. The cyanobacteria fixes atmospheric nitrogen into ammonia. Because of its nitrogen-fixing ability, Azolla has been used as a biofertilizer in many countries of Southeast Asia such as India, China and Vietnam and so many other countries. In China, it has been used to bolster agricultural productivity for over 1,000 years. Azolla is a genus of 7 species, and the species are Azolla microphylla, Azolla folliculoides, Azolla pinnata, Azolla caroliniana, Azolla nilotica, Azolla rubra and Azolla mexicana. As already said, the leaves of Azolla are bilobed, and the dorsal lobes, which are aerial and green, contain a specialized cavity that houses the nitrogen-fixing cyanobacteria Anabaena azollae. Mature cavities which are ellipsoidal with its interior surface covered by a mucilous layer. Several hairs, traditionally called as trichomes, protrude from the cavity surface into the mucilous layer, creating intimate contact between symbiotic partners, thereby enabling the exchange of metabolites. Anabaena azollae is a heterocystous filamentous symbiotic cyanobacteria that fixes atmospheric nitrogen into ammonia. The primary use of Azolla is its co-cultivation with rice. The most common practice is that before or right after rice planting, Azolla collected from the pond or from the nursery is dispersed in the field. In the first few weeks, thin canopies of the rice plants provide partial shade and plenty of sunlight for Azolla to grow. During this period, application of a small amount of nitrogen fertilizer and phosphorus stimulate the growth of both Azolla and Anabaena. During this period, Azolla is able to double its biomass in 2 to 5 days. Later in the rice growing season, overshading by the rice canopy and draining of water from the rice fields causes the death of Azolla, as a result of which the nitrogen which is fixed by Anabaena and the potassium and phosphorus which was assimilated by Azolla are released into the environment and are used for the formation of rice grain and for the next crop. In addition, Azolla is also used as a biofertilizer for other crops such as wheat, taro, banana and vegetables. For all other crops that require waterlogged condition, Azolla is used through intercropping. In some other cases, Azolla is grown as a monoculture is to be harvested and is plowed into the soil, after which crops are grown. In addition, Azolla is also used as a food supplement for domesticated animals such as pigs, cows, ducks, chicken and fish. Isolation and characterization of Anabaena azolla. First one is isolation steps. First step is sample preparation. In this step, azolla is grown in distilled water or in a nutrient deficient medium for a week. This nutrient deficient condition will induce the release of cyanobacterial filaments from the leaf cavities. In second step, the stressed fronds are then transferred into a flask containing a nutrient medium, typically nitrogen-free medium such as B11 or Venex medium. The flask is then incubated under continuous light about 2000 lux at a controlled temperature such as 25 plus or minus 2 degrees centigrade. After visible growth, the liquid culture is taken and plated on a agar nutrient plate containing B11 and is incubated. After incubation, the well-developed cyanobacterial colonies are taken out for characterization. Characterization method. Anabaena azolla can be characterized by different methods such as morphological analysis, physiological method and molecular method. Morphological analysis involves observing colony color, which should be brown to olive green in culture. Next is vegetative cells. Examination of Anabaena filaments under microscope to know about the size and shape of vegetative cells is also useful to confirm the identity of the Anabaena species. The vegetative cells should be cylindrical. Next is heterocyst. Heterocysts are specialized cells in which nitrogen fixation takes place. Heterocysts are usually spherical and they are mostly intercalary, but sometimes the cells can also be terminal. The last one is achinids. Achinids are also modified cells comparatively larger than vegetative cells, and these achinids are formed in series away from heterocyst, and these modified cells are cylindrical. So by examining all these morphological features, we can determine the identity of the particular species of Anabaena. In addition to morphological analysis, Anabaena can also be characterized by different physiological methods. For example, measuring nitrogenous activity and estimating chlorophyll content. Finally, molecular analysis Such a sequencing of 23S ribosomal RNA gene is also very useful to confirm the identity of Anabaena azolla. Cultivation of Azolla. First step is site preparation and tank setup. First of all, choose a suitable location and dig a pit or construct a tank, lining it with plastic sheet to prevent the loss of water. In next step, fine fertile soil is spreaded evenly across the bottom of the tank up to a depth of 5 to 10 centimeters. For growth of Azolla, suitable nutrients such as 1 to 1.5 kilogram of cow dung and 5 kilogram of single superphosphate is applied evenly per square meter. This is followed by the addition of water up to a height of 10 to 15 centimeters. Next step is planting and growth. Suitable amount of Azolla such as 200 to 500 gram is rubbed in hand and is applied evenly on the water surface. Azolla cultivation requires shading in order to protect the plant from intense sunlight. And as Azolla grow, nutrients are depleted. In order to replenish the nutrient level, cow dung and single superphosphate mixture are added weekly or twice a week. Third step is harvesting and maintenance. Azolla grows at a rapid rate, covering the surface in 7 to 10 days. In order to prevent overcrowding, 1 to 1.5 kilogram of Azolla can be harvested daily and can be used for different purposes. For pest control, if necessary, pesticides such as carbofuran can apply to the tank. Next, we will know about field application of Azolla. There are two different ways in which we can use Azolla as a biofertilizer to promote the growth of rice. First way is incorporation of Azolla in rice field prior to rice plantation. In addition to this, Azolla can also be applied to the rice field after transplantation of rice. Friends, this is all about Azolla and Anabaena. For more updates, subscribe my YouTube channel, Sam Biology Class.